Hey everyone, and welcome to Miss Estruch Biology and this entire math skills video for AS or A-level paper 1 AQA biology. Now this isn't literally every single math skill that is on the math skills section of the spec. Instead what I've done is gone through the specification, the theory part, so topics 1, 2, 3 and 4, and wherever they say in the spec, at this point this math skill could be assessed. That is what I've picked out and gone through for this entire video. So it's all of the topic related math skills that could come up for topics 1, 2, 3 and 4. And if you do want even more help on the math skills, then don't forget to check out my entire math skill workbook where it goes through every skill explained, modelled example, practice questions and the answers, which I'll link in the description and the QR code just here. And keep a lookout for the paper 2 A-level video that's coming and the entire set of math skills too. But for now that's it, let's jump into it. So let's go through the math skills that are specifically highlighted within the theory section for the AS or paper 1 for A-level biology. So here's the first math skill that comes up for topic 1 in the specification: calculating pH from hydrogen ion concentration using the formula. Now this exact math skill has never actually come up yet, but elements of it have, so principles around the log and also hydrogen ions, but literally calculating pH hasn't come up. But let's just go through how to calculate pH from the hydrogen ion concentration using the formula in case it does come up or principles of it do come up as well. First of all, pH is a measure of the hydrogen ion concentration in a solution. The more hydrogen ions there are, the lower the pH, and that's why acids, which release hydrogen ions, have pH values lower than 7. The formula that we use is pH equals minus log to the base 10 of the hydrogen ion concentration. The square brackets around H plus just mean concentration, and it must be in moles per decimeter cubed, which we can see here. Now the log function here is base 10, not natural log, so make sure you're pressing the correct button on your calculator. The minus sign is there so that a high hydrogen ion concentration gives a low pH, and a low hydrogen ion concentration gives a high pH. So that's what we mean by it flips the sense. It basically shows you high hydrogen ion concentration means low pH. So if we were to go through then an example, and imagine that the hydrogen ion concentration is 3.2 times 10 to the power of minus 5 moles per decimeter cubed. First you need to type that number into your calculator and then press the log button. You should get minus 4.4949. Remember, the formula says minus log so that we can take the negative of this value, which gives us positive 4.4949. Finally, we would round our answer. Because the concentration was given to us in 2 significant figures, we give the pH to 2 decimal places, and that makes the pH equal to 4.5. And here we can see it step by step, everything that you should get. You can also work backwards. If you know the pH, the hydrogen ion concentration is 10 to the power of minus pH. So for example, if pH is 7.35, the concentration works out as 4.47 times 10 to the minus 8. Next then we've got plot data from enzyme practicals in an appropriate graphical format. So the sorts of things you would need to do then is create a graph using the data that you're given or from your actual experiment, and you need to know what to plot. So your independent variable always goes on the x-axis, and that's actually the case for any graph. And the independent variable is what you are deliberately changing. Dependent variable always goes on the y-axis, and that is what you are measuring. You always need to make sure that you're using the correct units and you put the units on your axes. And for enzyme experiments, it should always be a line graph, not a bar chart. So here we can see an example of one, and here's some more just general graph rules that they'd be looking for if they were asking you to draw a graph in the exam. So they would give you a section of graph paper, And you need to make sure that you pick a scale so that you fill at least half of the graph paper, whether that's in an exam or in your required practical. You also need to make sure that your scale goes up by even increments. So you've got an evenly spaced scale. You should be plotting your graph with small crosses. So each data point, you put a cross for that position. And we should have this smooth curve line of best fit. And that could be a curve like we can see here, or you might actually get a straight line of best fit. Unlikely in an enzyme practical, because we do have this increase up to an optimum and then it either plateaus or it will decrease. And then lastly, we don't tend to join the line together dot to dot between those data points unless you have so few data points that you can't accurately predict the line of best fit. But in the exam, it's unlikely they would give that to you. That's more relevant if you did the experiment yourself and you only had 3 data points, for example. Next then is using a tangent to calculate the initial rate of reaction from a graph. So reaction rates, and this is all linking to the enzyme topic for topic 1. Reaction rates are often determined by analysing experimental data represented on a curved graph like we saw here. And when a reaction progresses, the rate is usually changing over time. So to measure the rate of reaction at a specific point, we have to use a tangent to the curve and calculate the gradient of the tangent rather than that line of best fit. So let's go through an example then. So step 1 is to draw and use your tangent, and we have to identify the point of interest. So in this case, it might be they want to know the rate of reaction at 6 minutes. So you need to find 6 minutes on your graph. Next then, you need to use a ruler to draw a straight line, which will be our tangent, that touches the curve at exactly the point that you chose. And then either side of where it touches, the gap between your tangent and the curve should have equal angles. So we can see here the gap between those two, that angle there and that angle there is equal. So that's how you know the angle or the position to place your ruler to then draw that tangent. Now we can find the gradient of the tangent. So choose 2 clear points on the tangent. It doesn't actually matter anywhere on the tangent where you do this because it's a straight line, so no matter where you do this on the tangent you'll get the same answer. But what we're going to work out is the change in y, which means if we did this point and this point, we'd be reading off the value here and reading off the value here on our y-axis and working out what's the difference between those, and we divide that between the change in x, so the time here, which is 0, and the time here, which is 9.6 minutes. And in fact, we can see all of that written here and worked out. So we then do the change in y divided by the change in x, which is our gradient, comes to 11.458, and here are our units: milligram per decimeter cubed, which came from the concentration, divided by time, which is why it's per minute. So we then go on to using base frequencies to calculate the proportion of other bases in complementary DNA strands. This is still from topic 1 and it's within the DNA section. So some key things just to be aware of to start with. DNA base pairing rules. Adenine is always going to bind with thymine. They're complementary base pairs. Cytosine will always bind with guanine. So that means in a double-stranded DNA molecule, whatever percentage of adenine bases you have, you will have the same percentage of thymine because they always have to pair opposite each other. And the same with cytosine. Whatever percentage of cytosine bases you have, that will be the same as the percentage of guanine. And because those are the only 4 bases you can have, the total of all of those percentages has to equal 100. So for this one, your method would be identify the percentage given in the question. So which of these bases do you have the percentage for? Use the base pairing rule to find out the complementary base, and then you would take that away from 100 and then half the rest between the other bases. But let me just show you an example of that. So we've got a DNA molecule contains 30% adenine bases. What are the proportion of the other 3 bases? Well, if we've got 30% adenine, we'll have 30% thymine. Then we know that adenine and thymine are half of the bases, the other half is cytosine and guanine. So if 30 is adenine, 30 is thymine, then those together add up to 60. Take that away from 100 gives us 40% left. That has to be equally split between cytosine and guanine, so they must be 20% each. Next skill then is using the magnification formula. So magnification is image size divided by actual size. And you could have to rearrange that formula to work out the actual size of an object as well. Unlikely they'd ask you to work out the image size because you would just look at the image and measure it with your ruler. So here's our formula again, and the size of the image is the size of the object in the actual microscope image, whereas the size of the real object or actual size is the real life size of the structure you're looking at, which is probably a cell or an organelle in the cell. So we've got an example here. If the size of an image under the microscope is 200 micrometers, the actual size of the objects is 50 micrometers, and the magnification is, this time we'd do 200 divided by 50, and that would tell us that the magnification is 4 times. So we can see just using that formula and the data that you were given. The units of measurement that you usually get are micrometers or nanometers for the actual size. And it's normally actually micrometers because most of the organelles in the cell are within the sizes that you would measure them in micrometers. We don't tend to use nanometers unless, like it says here, viruses are very, very small, so you might have the units in nanometers. But when you're actually measuring it on your image size, you're likely going to be measuring in millimeters, but they usually ask you to give your final answer in micrometers, so you do have to do that conversion as well, and we'll see that in our worked example. So if we have a look at this one, we've got what is the magnification of the mitochondrion in your image? Show you're working. So for this one, write out the formula first of all. We've got magnification equals size of image divided by the size of the real object. The length of the image, if we were to measure it, it's been measured to be 84 millimeters. And we've already been told that the actual length is 6 micrometers. So we'd need to do 84 divided by 6. However, they have to be in the same units. And 84 millimeters, you can fit 1000 micrometers into 1 millimeter. So if we have 84 millimeters, that means we have 84,000 micrometers. So whenever you're converting your image size, which will be in millimeters, into micrometers, you always times by 1,000. As we then substitute in those values and we get 14,000, that is our magnification. The next math skill we've got is calculating the mitotic index from microscope slides. And here's the formula that you need to know for this one. The mitotic index is the number of cells that you can see in your microscope image in mitosis divided by the total number of cells. So what this calculation basically is a measure of the proportion of cells undergoing mitosis, and it's used to estimate how quickly a tissue is growing, because if you have a higher mitotic index, that indicates more cells are undergoing mitosis and therefore there must be more growth happening. So here's our formula. If we have a look at how you'd actually calculate this, then you'd need to examine a microscope slide that shows dividing tissue. So for example, it might be an onion root tip, field of view from your required practical 2. You would then count the number of cells that you can see in any stage of mitosis, so whether it's prophase, metaphase, anaphase or telophase. Count the total number of cells you can find in that field of view, and then you'd use that formula, number of cells in mitosis divided by total number of cells. So just to show you an example here, we've got a field of view that shows us some onion root tip cells, and we can see, if you were to count them all, we've got 14 cells. 4 of them are in mitosis, so we do 4 divided by 14. Our mitotic index is 0.29. Now, a few things just to point out. First of all, only ever count the whole cells. So we can see here we've got a part of a cell, we've got a part of one there, a part there, there, and actually here as well. You wouldn't include those because if you can't see the whole cell, we can't actually see whether it's in mitosis or not. So only count the whole cells. And the next thing is, the way that we can tell if a cell is in mitosis or not is the chromosomes are visible. So this one here, we can see visible lines, which are the chromosomes. We can see visible lines in this one, and this one. Then in any particular organisation, so it's probably prophase, but we can see the chromosomes, whereas here you don't see any lines, you don't see any chromosomes. Now, the point I was making down here is, this is just an image that I've found online, but in the exam, they would give you an image where it was really obvious, where there was no debate about whether it is or is not in mitosis. It would be really obvious that you can see those lines. So just bear that in mind for the exam. Next we've got is using a scale bar to calculate the actual cell sizes. So in a magnified image, like we can see here, sometimes you actually get a scale bar, and from that you have to work out the magnification, and they don't give you the magnification. And in this one, we've got a mitochondrion, and we've been given this scale bar, and we're told that scale bar is 200 nanometers. Now, what that means is that line there represents the actual size of 200 nanometers. Not that if you were to look at that, that is literally 200 nanometers on the image. That is the actual size. So what we would then need to do is get our ruler and measure what is the image size of that line, because we know the actual size is representing 200 nanometers. And measuring this 16 millimeters, obviously it depends what device you're looking at, but let's say it's 16 millimeters. You could then use your image and your actual to work out the magnification. So 1 millimeter, again, we need to convert our units here. 1 millimeter is 1 million nanometers. So you would need to do times 1 million, because we've got 16 millimeters and there are 1 million nanometers in 1 millimeter. So we actually have 16 million nanometers. So we'd do 16 million divided by 200, and then that tells us the magnification of this image is 80,000 times. The next math skill is plotting results from permeability or osmosis experiments. And this could come up in your required practical 3 and 4, the ones where you're using potato plants for osmosis or the permeability of the beetroot membranes. So for the osmosis one, your experiment could be with plant tissue, such as potato cylinders, in different sucrose or salt concentrations, where you're going to be measuring a change in mass or length of your plant tissue. Then if you're plotting your results, if we go back to one of our earlier slides, we were talking about you always have your independent on the x-axis, your dependent on the y-axis. And the independent is what you're deliberately changing. And in this case, we'd be deliberately changing the concentration of the sucrose solution. So that would go on your x-axis. And we are measuring, our dependent variable, the percentage change in mass, or we'd actually measure the change in mass, so the initial and final mass, work out what the change in mass is, and then you always plot it as a percentage so it's comparable, taking into account the fact that they might not have been exactly the same mass at the start, and that goes on your y-axis. The permeability experiments, so this would be your beetroot discs, maybe at different temperatures or in different solvents or different concentrations of solvent. And what you'd be measuring is how much pigment is in the solution after a set amount of time. And you could do that using a colorimeter, so you get a numerical value or quantitative rather than just subjective, describing how dark the purple colour of the pigment from beetroot is. So in terms of plotting the data, our independent variable depends what's actually being varied. It might be the temperature, it might be the type of solvent, it might be the concentration of the solvent, and that goes on your x-axis. The dependent variable is what you're measuring, and we are measuring the amount of light being absorbed in that colorimeter. Or sometimes it's measured as the amount of light that transmits through. I always go for absorbance. So thinking about our graph rules, it would be a line graph because it's continuous data. We have to label the axes and we have to give the units as well. You have to use a suitable scale, meaning your graph fits at least half of the paper, and you have to increase by equal amounts on your scale. And lastly, plotting your data points with an X, and then you draw a line of best fit, which is likely to be a smooth curve. So here's some example data. We've got our sucrose concentration, and we've got the percentage change in mass. And here is our graphs. We've got percentage change in mass, sucrose concentration, and there's our curved line of best fit. And this actually links to the next math skill, determining the water potential from the intercept on a graph. So using that same concept, we've got our sucrose concentration, percentage change in mass, and we've got our data plotted. The intercept, so this is where if we were to draw a dashed line at where we have 0 change in mass on our y-axis, that would be the water potential inside of the plant tissue, which in this case is potato. And the reason for that is, if you don't have any change in mass in an osmosis experiment, that means there's been no net movement of water into the potato plant or out of the potato plant. And if there's no net movement of water by osmosis, that tells us that the water potential inside of the potato tissue must be the same as the water potential of the sucrose solution. And that then indicates to us that at whatever point the concentration is that we intercept at 0, that must mean that's the same concentration inside of the potato. For instance, it tells us that the potato tissue must have a sucrose concentration of 0.4 because that is where the concentration is. Now, if you wanted to know what that was as a water potential, it's not literally just 0.4 because that's a sucrose concentration, not a water potential concentration. So sometimes there's an extra mark for saying you then need to look up in a table to see what water potential that concentration correlated to. The next skill, calculate the surface area to volume ratios of simple objects. So for example, cubes. So why does this matter? This is now topic 3. Small organisms and cells exchange substances faster. So it links to the topic to do with gas exchange and absorption. Surface area to volume ratio shows how efficient an exchange surface is. So if you have a high surface area compared to the volume, or surface area to volume ratio, that means you'll have faster diffusion or faster exchange relative to the size. And that could be diffusion of gases, absorption of substances, it could even be loss of heat across the surface. And if you have a lower surface area to volume ratio, you have a slower exchange system. And that is then when you start to see organisms have evolved over time and have these adaptations to increase exchange, even though the entire organism has a low surface area to volume ratio. So, for example, gas exchange in the alveoli in the lungs, or gas exchange in gills. So the formula for a cube would be working out the surface area, which is 6 times, because there's 6 faces of a cube, the length squared, because the area of one side of the cube is the length times the length, and then there's 6 faces, so times 6. The volume of a cube would be the length cubed, or in other words, the height times the width times the length, but because it's a cube, those will all be the same dimension. And then it's whatever the surface area was divided by the volume. And we always present that as whatever the answer is to that calculation, we have that value to 1. So it's a ratio still. So we've got an example here, a cube with dimensions of 1 centimetre. So 1 centimetre in height, length and width. If we were to do our surface area, the length squared is still 1 times 6, because there's 6 faces to that cube. So that gives us 6 centimetres squared. The volume is the length cubed. So 1 times 1 times 1, which is 1. So 1 centimetres cubed. So that means our surface area divided by volume is 6 divided by 1. So we have a ratio of 6 to 1. Next then, we have rearrange and use the formula for pulmonary ventilation rate, which is pulmonary ventilation rate, or PVR, is tidal volume times breathing rate. So the PVR, the pulmonary ventilation rate, that is the volume of air ventilated per minute, and that would typically be in decimeters cubed as our unit for volume per minute. Tidal volume is the volume of air per breath, and because it's volume, it's going to be in decimeters cubed. And then breathing rate is number of breaths per minute. So that would be our formula. To rearrange it, if you wanted to find the tidal volume, it'd be your pulmonary ventilation rate divided by breathing rate. To work out the breathing rate, it would be pulmonary ventilation rate divided by the tidal volume. So we've got a few examples then. A person has a tidal volume of 0.5 decimeters cubed and a breathing rate of 12 breaths per minute. What is the PVR? So this time it'd be 0.5 times 12, and that tells us the PVR is 6 decimeters cubed per minute. Another example, we've got a person has a PVR of 7.2 decimeters cubed per minute and a breathing rate of 18 breaths per minute. What's the tidal volume? So this time would be 7.2, so PVR, divided by the breaths per minute, 18. So the volume, the tidal volume is 0.4 decimeters cubed. Last option then, a person has a tidal volume of 0.6 decimeters cubed and a PVR of 9 decimeters cubed per minute. The breathing rate would be 9 for PVR divided by the tidal volume, which is 0.6, so they have 15 breaths per minute. Math skill 12 on this list is from topic 4, using 2 to the power of n to calculate the number of chromosome combinations. And this links to meiosis, where n is the number of pairs of chromosomes and the number of homologous pairs. So the principle here is that during meiosis, chromosomes line up at the equator in independent segregation and then separate apart. And which side of the equator the maternal and paternal chromosome align is random. So each homologous pair can arrange in 2 different ways, either on the left or the right of the equator. So if we want to know how many possible different combinations there are that we could get in the gametes, it's 2 because we have pairs of chromosomes to the power of n, where n is the number of pairs you have, the number of homologous pairs. And in humans, we have 23 homologous pairs of chromosomes. So that means 2 to the power of 23, which comes to over 8 million possible gamete combinations that we can create before you even take into account crossing over and random fertilization of these gametes. So it's another way just to demonstrate how large the variation, genetic variation is that's introduced from meiosis with a mathematical formula. Next then we've got interpret data using logarithmic scales, and this often links to aseptic technique and growing bacteria because we use a log scale when you have a really large range of data. So many biological processes involve exponential growth and decay. So for example, bacteria, replication, but we can also actually see this in the growth of cancer cells. It comes up quite a bit in paper 2, the same skill. So logarithms help compare values that have a very large range, so comparing very small values to very large ones. And we can have logarithm to the base 10 or log 10, and that is used for bacterial growth and dilution calculations. Natural logarithm, or ln, that's mainly what we see, but you can get log e as well, is applied in growth rate equations, but they'd normally give you an equation that you'd need to use if you had to do that. So just a bit more information on this. Here we can see some original values, and this could be number of bacteria in a solution after a certain amount of time. And we can see here we've got data that is very, very small going up to very, very large. And it'd be difficult to plot this on a graph so that you can actually get a scale that you could fit all of this on. And actually, this probably isn't data from the number of bacteria because that's not a whole number, and nor is that. And you'd have to have a whole number for number of bacteria. But it's demonstrating that concept. You've got a really, really large range. So instead, you'd have to convert your raw original values into log values, log 10. So we can see here we've converted to log 10, and what that means is if you present your original value in the order of magnitude value instead, and this then gives us a much smaller range, and we can then plot that on a graph. Now you can actually also get graph paper, which is logarithmic graph paper, and we can see here in this graph that's what we have. You don't have even subdivisions on this graph, and it's important that you know what each of those subdivisions means and how to read it off. So we can see here that where we've got 10 to the 2, and that is the first line at 10 to the 2, because we've then got 1, 2, 3, 4, 5, and it actually goes up 10 divisions. The first line, the first point on 10 to the 2, what that means is 10 to the 2 times 1, which is 100. This here is now the third line up, because we've got, that was line 1, line 2, line 3. So that means it's 10 to the 2 times 3, which is 300. This one here, we've got 10 to the 4, that's the first line, second, third, fourth, fifth, So that's 10 to the 4 times 5, and so on and so on. So that's how you read off on the y-axis on one of these graphs. So each of those lines is representing times whatever number line that is. Number 14 on our list is use and interpret the index of diversity formula, which is from topic 4. So here is the formula. They usually give this to you in the exam, but they don't often tell you what each component means. Sometimes they do, but they don't always. So D is index of diversity, and this is a measure of biodiversity, taking into account species richness and also the number of individuals within each of the populations of species. Capital N is the total number of organisms, so every living thing in that community. Lowercase n is the number of organisms of just one species, and that's why we have this symbol here, sum of, because this part you have to do for every species in your community, and then you add up all of those for all of your species, and that's normally where people go wrong on this formula. But if we go through some examples. First of all, why do we actually do this? As I said, it's a measure of biodiversity in the community. The higher the index of diversity, or D, that tells us we've got a greater biodiversity, which means we've got more species and more balanced population sizes in the species, meaning each species probably has a large population. A lower index of diversity means lower biodiversity. So you might have fewer species, or you might just have one species that dominates. So you might have 10 species present, but you've got 1 million individuals in one of those species and only 5 or 2 or a very low number in all of the others, and therefore they're at risk of going extinct. So it's not actually a very good, high biodiversity. So here's an example. They wouldn't give you this column in the exam. They don't make it that easy. They tend to say, here's your species, here's the population size. So capital N is the total number of individuals in this community. So we'd need to add up that column, and that comes to 50. For this bit, we need to do the sum of lowercase n times lowercase n minus 1. So we have to do that for all of the species present. And as I said, they don't give you that column, so I just tend to add it on in the exam and draw it on myself. So this would be 20 times 20 minus 1. So 20 times 19. This would be 15 times 14. This is 10 times 9, and this is 5 times 4. So you're always doing n times n minus 1. And then it's the sum of that column, and that comes to 700. So this numerator here then is the total number of individuals, which was 50, times 50 minus 1. So that is 50 times 49, which is 2,450, divided by our denominator, which was the sum of lowercase n times lowercase n minus 1. And it was the sum of all of those, which was 700. So we've got an index of diversity overall value of 3.5. Now, one thing just to try and gauge whether you've done this correctly, using this exact formula, you usually get an answer between 1 and 10. So if it's drastically over 10, you've gone wrong somewhere. And if it's lower than 1, you've probably gone wrong. If it's lower than 0, you've definitely gone wrong. Next then, calculating mean values for a data set. So this was quite straightforward. Mean is a measure of an average. So it's the sum of all the values divided by the number of values you have. So add up all of your repeats, divide by the number of repeats you had. So we've got an example here. Here's all the repeats. Add them all up, and there were 5 repeats, so we divide by 5. We've got a mean of 9. Now this goes hand in hand with standard deviation, and although it says on the spec calculate standard deviation, you might be asked to do that in your required practicals, but they won't ask you to do it in the exam because they do state it'd take up too many marks because of the time you'd need for it for the number of maths marks you're allowed. But you do interpret standard deviation, and that comes up every single year. So when we're doing an experiment and we've collected the data, we usually calculate a mean, but it's important to do more than just calculate your average or mean. It's better to also do the standard deviation because that tells us how spread out all of those results are compared to your mean. So it gives us a better idea of the reliability, how spread out all of those data points are. So standard deviation compared to the range of data is better as well, because the range just tells you the highest and the lowest. Standard deviation is considering all of your repeats, how spread out are they compared to the mean? So when calculating standard deviation, one standard deviation is taken into account 68% of the data, and 2 times whatever your standard deviation is includes 95% of the data. And they normally give you that as a statement in the exam. They'll say standard deviation has been included, and it's 2 times the standard deviation, which includes 95% of the data. Now, we don't need to get into the nitty-gritty of the maths behind that. What you need to know is that means for you that when you take into account your standard deviations against the mean, if those values overlap, it tells us you do not have a significant difference between those two means. So that is the point of telling you that fact. So just a few other points that we've got here. The standard deviation shows the spread of data around the mean, like we said, whereas the range only shows you the highest and the lowest value. So it's not as useful. The standard deviation reduces the effect of anomalies because it's taken into account all of the data, whereas the range includes the anomalies because it's just showing you the highest and the lowest, so it can be quite skewed. And the standard deviation, this is the big thing that it's used for. It's used to indicate whether a difference between mean sets of data sets is significant or not. That's the main thing that is assessed on in the exam every year. So let's just focus on that and have a look. We've got an example of some data here, 2 different types of treatment, mean population size, here's our mean, and we're told in brackets this is 2 times the standard deviation. So what you would need to do is, from our highest value, highest mean is 7.3, we'd need to minus 0.8. From our lower, which is 5.6, we need to add on 0.7, and then see 5.6 plus 0.7, does that take the mean higher than 7.3 minus 0.8? And if it does, that means the standard deviations overlap. And if it doesn't, then that means they don't overlap. If you have an overlap, that would mean there is no significant difference between the means. Even though those numbers are different, 5.6 and 7.3 might be different. When you take into account standard deviations, they're not significantly different. You could also get it on a graph, which is easier to interpret because you can then just visibly see, does this standard deviation line cross the same point on the y-axis as this one? And we can see here it doesn't. So that means we can visibly see those means are different. The standard deviation bars also don't overlap, so it tells us that there is a significant difference between those means. So hopefully you found this helpful. If you did find this helpful, then in my AQA math skill workbook, I have got every single math skill that is listed in the math skills section of the specification in this workbook. You have an explanation of every math skill, a modelled example, practice questions, and all of the answers. And I'll link that in the description. But that is it for today's video. Look out for the paper 2 version and the entire set of skills still to come.